So here's our second part in the photosynthesis lecture. And again, things I want you guys to remember. The key structure here is a chloroplast. That is the structure that enables an organism to run photosynthesis. Generally or generically, we call anything that runs photosynthesis a producer. Plants are an example of a producer, but there's also algae and bacteria and other organisms that can be classified as producers because they have the chloroplast. So chloroplast is one of the main things that you need. The other main thing, or one of the other main things that is needed, is sunlight. And the more sunlight that the chloroplast can capture, the more energy it has to run photosynthesis. So take a look at the spectrum of light. Light travels in wavelengths, like waves in the ocean, and they have different heights, different sizes to them. Visible light is light that is used for photosynthesis. Now, visible light ranges from 400 nanometers all the way up to 740 nanometers. This is what gives us our color spectrum. So think about red, orange, yellow, blue, green, indigo, violet, the colors of the color spectrum, or the colors of a rainbow, or I have a little prism down there. Light passes through and it reflects out all these different colors. That is what is used for photosynthesis. So those wavelengths of light have energy to them. And as they travel from the sun, or a light source, to the chloroplast, or any structure that can capture those wavelengths, they travel in different sizes. So if you can capture all of these, you're incredibly efficient because you can get all that energy from that light and use it for your process of photosynthesis. If a bunch of these bounce back, then you're not very efficient. So when we look at plants and we look at chloroplasts contain the pigments chlorophyll. And there's a chlorophyll A and a chlorophyll B. Those are the primary pigment molecules found in the chloroplast. What bounces back from the chloroplast is the actual color that we're going to see, that is visible to our eyes, the color and the color spectrum that we can see. So leaves typically appear green because chlorophyll cannot capture the green wavelengths of light. So the analogy here, imagine a baseball player, a catcher, behind home plate, has a certain size glove, and that glove can catch certain sized balls that are being thrown at him. The balls that bounce out, he can't catch. He can't grab that energy. So when it comes to chlorophyll A and chlorophyll B, they can catch all the balls that are purple and blue and yellows and oranges and reds and those colors, but they can't capture the balls that are the green color. The middle wavelengths are about 550 nanometers to about 600. That is what bounces out. That is what the chlorophyll pigments can't capture, and that's why we see green, because it's bouncing out of the chlorophyll. Now, the other pigment, or the other structure that can also run photosynthesis, are called chromoplasts. Now, chromoplasts contain a pigment called the carotenoids. This is your reds, your yellows, your oranges, your fall colors. Carotenoids are just like the baseball player with the glove, but they can only catch blues and the dark purples and all the wavelengths on the left-hand side of the color spectrum. Carotenoids cannot capture anything in the reds, the yellows, the oranges. They really don't do a very good job with the greens either. So those are the colors that bounce back to us when we look at the carotenoids. Now, generally, when we look at a plant and we see a leaf, whether it's an oak leaf or a maple leaf or whatever it may be, we see a lot of green. It's full of green. So here's the oak leaf in summer, solid green, because you're actually looking at a massive number of chloroplasts containing chlorophyll, the green pigment, And that green is bouncing back because they can't capture those wavelengths. So that's why the leaves look green. But consider as we move into fall. As we progress in the fall, chlorophyll starts to break down, starts to degrade. It's not being replaced. So there's fewer and fewer of the green pigments, fewer and fewer of the chloroplasts within the leaf, and the colors start to change. And we start to see reds or yellows or oranges or these things we call the fall colors of an oak leaf. So the bottom leaves are kind of a reddish orange color because the chromoplasts are now visible. Yeah, they've been there the whole time. They've been there all along. They just have been masked by the green chloroplasts. So if you had, and I'll make up some easy numbers here, if you had 1 billion chloroplasts that are green and 100,000 chromoplasts that are red, all in the leaf at the same time, you're going to see primarily green because they just override the other colors. There's just so many of the green ones. But later in the fall, all those green ones start to slowly disappear and break down and degrade, and their numbers drop dramatically. Yet the chromoplasts remain. And those continue to enable the plant to photosynthesize throughout some parts of the fall, but eventually even the chromoplasts will break down, they will degrade, and the plant leaf will fall off the tree, and the plant will move into a complete period of dormancy here. Okay, so this is the pathway. This is what we're going to be looking at as we explore photosynthesis. Now, there are actually 2 types of photosynthesis. We talked about the anoxic. This is the old system of photosynthesis. This is what's known as the cyclic system. So let me add that in here. Cyclic system is also referred to as the old system. The system, TEM. All right, so this system evolved billions of years ago. And when we look at it, it's a circle. It's a circular system. The only thing still using this are those bacteria we talked about earlier in the first part of the lecture series here. So the way this will work, energy from the sunlight, that's this little zigzag lightning bolt looking structure, a photon of light hits the chlorophyll, hits the pigment. That excites this thing called the reaction center. And when that reaction center gets excited, it pops out the electrons. The electrons shoot up. They're up here in an excited reaction center. They get transferred to an electron acceptor. The electron acceptor literally is a structure that carries the electrons. It moves it over to this BC1 complex, and this is where ATP is generated because of the electron. The acceptor carries the electron back to the photosystem, drops it off right here, and says, do it again. Bang, up, down, make ATP, back to the beginning. Bang, up, down, makes ATP, back to the center, do it again. This is why we call it cyclic. Circle. Now, seems like a good concept, but it's limited. So consider making up some easy numbers. If you had 100 electrons in your system, and you bounce them all up one at a time, 1, 2, 3, 4, all the way through the 100 electrons, they're all going to go up here, but sometimes they get lost, and all of a sudden, whoop, there goes an electron, shot off screen. So now maybe you're down to 95, and the acceptor drops a few. Oh, now you're down to 88. And they go through here, make an ATP, the acceptor brings, it lost a few more. Maybe it brings 82 of them back to the photosystem. And as this keeps going through the circle, your electron numbers get depleted slowly over time. Eventually, your cyclic system will not be able to generate ATP. That's a huge problem. Another problem is that this system is completely dependent upon sunlight. Not that the other systems don't use it, but when this loses sunlight, everything stops. The system shuts down. It does not continue without a constant influx of light. So there's no backup energy for the organism running this pathway. That can be a bad thing. That can be a problem. Okay, so again, here's our old cyclic system. Now, early on Earth, when there was very little life, there was very little competition, this system would have been perfect. It would have been completely functional, worked fine, no concerns. But as life continues to adapt and evolve and compete, we have to improve upon the system. The original design is solid, but as producers continued to evolve because of natural selection, they had to, again, modify and improve upon this system. So that's what we have today on Earth in the non-cyclic system. This is the newer one. This is an improvement upon the old system. A lot of the basic fundamentals are there, but with some add-ons and some modifications. Now, the non-cyclic system is composed of two parts. Part 1 is light dependent, and then part 2 is light independent. Okay, so it's got a two-part system here. Part 1, part 2, light dependent, light independent. And what we're going to see when they go through this pathway, this non-cyclic system, it has modified, in a sense, changed, tweaked, adjusted the flaws we saw in the cyclic system. There's a constant replacement of electrons, and there's a backup storage of energy produced here. All right, so let's take a look at how this part 1 light dependent works. Now, specifically, this is going to work at the chromoplast or the chloroplast, at the structure where it can capture sunlight, and it's going to occur in this little green stack of pigments known as the grana. Now, grana looks like a little stack of green coins here. There's our little grana. Let's put that term in there because that's going to be an important term for you guys to remember. Grana, and I'll give you a little arrow that will show us where it's at. That little stack of green coins. Okay, so those guys are responsible for capturing the sunlight down here. So they capture the sunlight. They get all excited. Electrons pop up, get caught by an electron acceptor. They move down the electron transport system. That's the same type of system we saw back with cellular respiration, a series of proteins. And as those electrons move down this electron transport system, they generate ATP. Awesome. That's exactly what we need, ATP. They go down to this second group of pigments. And silly trivia information, photosystem 2 versus photosystem 1, they are named that way because this one was the first one discovered, then scientists discovered this one. Two guys, Calvin and Benson, are the ones who did a lot of work with this. Okay, so anyway, trivia aside, electrons come down to photosystem 1. They get excited again by the sun that they're being exposed to. Pow! Shoot them up. They get caught by an electron acceptor, and they get placed into our little wheelbarrow, the NADPH. Those electrons add or attach the hydrogens into the wheelbarrow. Now, they're only going in one direction. So electrons shoot up, boop, click all the way down the steps, doop, 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 shoot up again, boop, and dump right into the wheelbarrow. Then they're gone. So in order to continuously replace them, we have to have water available for the plant. The water breaks apart. Electrons get released, and they will diffuse into the photosystem here to then shoot up, drop down, shoot up, and go into the wheelbarrow.